All right, welcome to the second lecture of the first week of the fall semester 2020. This chapter is much bigger than the first one, and I do have a lot of material to go through. Just a reminder that you should be taking notes. You can pause the video at any time so that you can take notes, and I try to review the closed captioning so that if you need help spelling anything that I'm saying, that those corrections should be there. It's not perfect, so if you catch something that's incorrect in the captioning, do let me know, and I can go back and edit that and make sure that I have the best captioning possible, both for everyone so that they can see and hear what I'm saying, but for anyone who needs that closed captioning, I want to make sure that it's as good as it possibly can be. So Chapter 3 is covering cells. We're obviously not going to be able to cover all the details of all types of cells, but just an introduction to the two main categories of cells that we'll be working with throughout this semester. So we've got, as we mentioned last time, 3 domains. We have the bacteria, we have the archaea, and we have the eukarya. We've got eukaryotic giardia. We saw that in the first lecture. We have some prokaryotic bacteria that are bacillus. And image C, we've got some very large by microscopic sizes. That's 35 microns, so that is a eukaryotic microbe. And D, tremendously large, 40 microns. Typical bacterium, less than 2 microns. So this is a eukaryote. Looks like it's probably a paramecium. These are red blood cells, and this is a parasite, a eukaryotic parasite that has infected someone's bloodstream. And finally, we have some spirillum, those bacterium that have that tight corkscrew appearance. So when we're getting into cells, knowing that cells existed, something microscopic that we couldn't see with our naked eyes, but could be so important to our functioning and to our health. Originally, it was thought that life was somehow more special than non-living things in terms of its composition. Now, clearly, living things are distinct from non-living things. We have some great features that non-living things do not. But it was thought that living things could generate spontaneously, meaning that living things could arise from non-living matter. And there are several experiments that were done to try to prove or disprove that. So one of the early thoughts was that flies, or maggots, came from meat. Not that they landed on it, but that the meat actually produced those. So if you leave meat in an open container, yeah, flies land on it, they lay eggs. Baby flies are actually maggots, and that clearly happens. And Dr. Reddy put a cork in there, and no maggots. The criticism of his experiment was that air is vital to life, and if you shut off the air, well then obviously the meat could not generate the life. So he said, fine. Covered it so that air could get through. If maggots showed up, they were on the outside, and they were not on the meat. Louis Pasteur took that further to look at the generation of even microscopic life, and had flasks that he heated the neck to make it, what they call it, swan-necked, because it had a couple of crooks in it. So the end is open. Air can get into that. The broth that's in there, he heated until boiling for several minutes to make sure that it was sterile. And left on a bench top for a very long time. Nothing ever grew. The liquid in the bottom of the flask remains clear. Some of those flasks still exist in Dr. Pasteur's lab in Paris. I got to see those on a trip. And periodically they do tilt the liquid and then check, because dust can get in there, and then shows that that broth, even more than 100 years later, is still capable of supporting life. But unless living things land in that broth remains sterile. Robert Hooke was the first to show cells. He used cork, and they were called cells because the Latin word cellulae means little rooms. and they look like little rooms. So the cell theory is that all living things are made of one or more cells, and that all cells come from pre-existing cells. Now that doesn't cover where did the first cell come from. That has to do with the idea that originally there were chemicals such as RNA that eventually coalesced with some fat around them that made a cell. But now that we have cells on Earth, all cells come from other cells. The endosymbiotic theory, endo means inside, and symbios means living together, because bio is life, and to be SYM is together. So it's the theory that there was things living inside of each other. And in particular, this is talking about how eukaryotic cells evolved. That there was some sort of large proto-eukaryote. In the first event, it consumed an aerobic, meaning an organism that requires oxygen. That bacteria was engulfed by this original prokaryote with its DNA, and then that eventually became the mitochondria. And in a second event, some of these proto-eukaryotes engulfed a photosynthetic bacterium that now are the modern-day eukaryotes. And there's a lot of evidence for this, not just the size of the mitochondria and the chloroplasts, but both mitochondria and chloroplasts have their own DNA. They have their own ribosomes, so they do their own protein synthesis, and both mitochondria and chloroplasts are capable of dividing independent of the cells that they're in. So cells that contain those organelles, if there's higher energy demands, then the mitochondria and cells that contain them and the chloroplasts and cells that contain them divide to meet that increased energy demand. Joseph Lister, first glance, maybe I've never seen this guy, maybe I've never heard of him. Proper care of surgical wounds and sterilization of surgical equipment, and also is why the mouthwash Listerine got its name, because that helps to kill the bacteria that could cause gum disease in your mouth. And there's Robert Koch with his cell theory. Now this is a generic prokaryotic cell. Pro means before, and karyo, K-A-R-Y-O, means kernel or center. So it's before the kernel, before a nucleus. So it is a cell. There is DNA. The nucleoid is wherever the DNA is sitting in a prokaryotic cell. And this cell happens to be kind of evenly distributed, but it could be clumped down here, it could be over there. Wherever it is, that is the nucleoid. Plasmid is a circular piece of DNA that is distinct from the chromosome, typically encodes features that are not critical to the life of the bacterium, but give it some advantages, like antibiotic resistance or being able to grow at a higher temperature. All cells have cytoplasm, that is the semi-fluid liquid inside the cell. All cells have a plasma membrane, also called the cytoplasmic membrane, also called the cell membrane, but the membrane. Almost all prokaryotic cells have a cell wall. Many of them have a capsule. We're going to be doing or observing stains that are specific to help us see the cell wall composition and to help us see whether or not the microbes have a capsule. The cell wall is for protection. The capsule is a gooey, sticky layer outside that cell. The fimbriae are these projections that lead out. They're attached to the plasma membrane, go through the cell wall and the capsule. They help the organism with attachment. And we also have inclusions. Prokaryotic cells don't have any internal membranes. They have just the one plasma membrane. So inclusions are concentrations of necessary chemicals. The pilus, much longer projection that's coming attached to that plasma membrane through the wall and the capsule. Also for attachment, can stick cells together for genetic exchange as well. And lastly, we have a flagellum. If we had more than one, there would be flagella. And you might guess that because it is so long that it is used for movement, and you'd be correct. So there are pictures of the various shapes of microbes. Again, we have the coccus, the bacillus, the vibrio, coccobacillus, spirillum, and spirochete. And there are examples of those in both a drawing and electron micrograph. We also have arrangements. When we observe our stained slides, you're going to be asked to identify the cellular morphology, the shape of individual cells, but also the cellular arrangement. How, if at all, do they clump together on a slide? So you could have a single coccus, you could have diplo, di meaning pairs, you could have tetrads where you have fours, strepto, chains, staphylo, clusters. And you could have the similar descriptions for bacillus. You could have singles, you could have streptobacillus, you can have staphylobacillus. When we look at how cells survive in their environment, the tonicity, T-O-N-I-C-I-T-Y, tonicity of the solution can matter. We can have an isotonic. The tonic refers to the relative proportion of particles in a solution surrounding the cell versus the interior of the cell. And iso means the same. So in this case, we have a 20% solute concentration outside the cell. We have a 20% solution inside the cell. And water can move in and out of the cell, but will do so equally. One molecule out, one water molecule in. And a hypertonic means too much. The higher solute concentration. And you do have to say which one is hypertonic. So in this case, the exterior environment is hypertonic to the interior environment. And in order to balance that, to try to get the same tonicity to become isotonic, water would move out of the cell. And we're doing that because the particles are shown as being too large. If the particles were very, very tiny or nonpolar, they might be able to freely diffuse across that membrane. But if they're too large, they cannot. And the only way a cell can try to achieve a balance is to move the water so that there's the same proportion of water to solute. So if the solution outside the cell is hypertonic, water will move out. We have a high concentration of water inside the cell, a low relative concentration of water outside the cell. Water will move from a high concentration of water to a low concentration of water. In a hypotonic solution, the solution outside the cell has too little, hypo, under, like a hypodermic needle goes under your skin. So hypotonic, we have too little particles. We have more particles inside the cell. So water will move from where there's a lot of water outside the cell to where there's little water inside the cell to try to maintain a balance. And if you look at this little cartoon cell, when it's isotonic, it's nice and round, it's happy. When it's in a hypertonic solution, water moves out and it shrivels up. And if it's in a hypotonic solution, water moves in and it will swell up. If it has a cell wall, that might protect it. If it does not, that cell will burst. So in a prokaryotic cell, they have a cell wall primarily. Isotonic, water moving in and out, everybody's happy. Hypertonic, right? The water is moving to where there's less water, lots of water inside, less water outside. Cell membrane shrivels, and we get plasmolysis. And in a hypotonic, the water moves in, and we get swelling. The cell may not burst, but the water moving in might dilute the chemicals that are necessary inside that cell and decrease its functioning. So here is a really terrible image of an electron micrograph of a prokaryotic cell. They're outlining it, showing this is the region where the DNA is sitting. and that's the nucleoid. That OID suffix tells you that it's kind of like, but not really. So it's not a nucleus, there's no membrane around it. It's like a nucleus, only in that that's where the DNA is sitting. Ribosomes are made of RNA and protein. There is no membrane around them, but they associate to make sort of a clump of a molecule. So there's a small subunit, and there is a large subunit. This is prokaryotic. You don't have to memorize the sizes of them. The S stands for Svedberg units, and that has to do with if we were to put ribosomes in a centrifuge and spin them. The Svedberg unit tells you how far down in a test tube they would settle, and that's based on their weight, like how big are they, but also their shape. So two things could have the same weight, but different shapes, they would have different Svedberg units. Because if you add 30 and 50, you would expect 80. Instead, you get 70. Because you've got one subunit here, you've got another one underneath, and when they come together, you lose some surface area. So the surface means the Svedbergs of the individual units don't add up to the same. The numbers are not what's critical. Is that there's a small and a large subunit. They are made out of RNA and protein, and they come together to be the complete ribosome. And there are different types of inclusions. They might be lipid droplets, they might be volutin granules, sulfur granules, magnetosomes. Some prokaryotes actually have chemicals that allow them to magnetically attract iron as a nutrient. And so there's lots of different ways that prokaryotes can concentrate chemicals that they need. Sporulation, the formation of spores, is very important because the spores are a way of preserving genetic material when the cell is in harsh conditions. And those harsh conditions could be lack of water, lack of nutrients, too high of a temperature, too cold of a temperature, anything that is stressful and would not allow the cell to do its normal cell division. So forming the spore, the DNA replicates, so we get a copy of it. A membrane forms around that copy of the DNA. We get more membranes, and then we get a cortex, which is a protective layer of carbohydrates and proteins, and eventually that is released. And so the cell would die, the DNA that was part of that cell is destroyed, but the spore is extremely resistant to heat and temperature, cold types of temperature, lack of water, too much water. It is just a little time capsule waiting for the right environment. So we will see this type of micrograph in lab. This is bacillus, and the cells will stain red, and the endospores are green. So we still have some living cells in this micrograph. We can see some cells where the endospores are still inside, which would be this stage. But we also see a few endospores that have been released. The plasma membrane, or the cell membrane of bacteria and other prokaryotes, is structured very similarly to eukaryotes in that it is that phospholipid bilayer. So the phospho are the phosphates, the lipids are the fatty acid chains, bilayer because there are two layers. But in prokaryotic cells, there are some special features that we may not have in our cells. We do have protein channels. We have peripheral proteins that are only on one side or the other of the membrane. Channels go all the way through. We might have glycoproteins, but the proteins themselves are very specific to the cell type. So we saw a little bit about water moving across a membrane, but we might also get the solute itself moving. So if we have that phospholipid bilayer, that is the innermost layer. That's going to be what controls what's coming in and out of the cell. Here we have the extracellular fluid is very hypertonic compared to the interior of the cell. So water would probably move from where there's a lot of water by osmosis out to the extracellular fluid, but these molecules are small enough that they're able to do simple diffusion from a high concentration of the solute particles toward a low concentration of the solute particles, and would do that until we reached a state of being isotonic. At that point, that doesn't say the solutes won't move at all. It just says that if one goes across to the interior, one will go across to the exterior, and water would also be moving in a balanced fashion. Facilitated diffusion means we've got difference in concentration, but the molecules are too large or too polar to move on their own, and so we have a protein channel that will facilitate that movement. So there's no energy required for the particles to move. The energy was required in building that protein, but once the protein is there, no further energy for the molecules to cross. Active transport is going against a gradient. So we have to provide energy. We are going to see this molecule, ATP, a lot. It stands for adenosine triphosphate. And when pieces of it, the phosphates, break off, that releases an enormous amount of energy that allows us to do a lot of different types of cellular work. We see on the right that in the outside of the cell, there's a high concentration of sodium and a low concentration of potassium. On the inside of the cell, in the cytoplasm, there's a low concentration of sodium and a high concentration of potassium. And even though we could, theoretically, by simple diffusion, move those, we do not because they are charged. Sodium ions and potassium ions are positively charged, and charged ions do not freely move across. And they don't go by facilitated diffusion because we are using and maintaining this difference in the concentration of sodium and potassium to do other types of cell work. So we load 3 sodium ions into the channel. The ATP energy releases them to the exterior. That opens that channel to the exterior, and 2 potassiums can bind and they get imported into the interior, maintaining a high concentration of potassium inside and a high concentration of sodium outside. So this is a cartoon of bacterial cell walls. We have something called gram-positive and gram-negative. Gram because Dr. Gram figured this out. Positive and negative just mean positive, it has the structure we're looking for. Negative, it doesn't have the structure we're looking for. We're going to have positive and negative results for every single test that we do in lab. And every single test is independent of the gram reaction. So just because something is gram positive doesn't mean it's positive for other things. Just because it's gram negative doesn't mean it's negative for other things. But it's called gram positive because during a gram stain that involves adding a dye, doing a decolorizing step, and adding a second dye, the first dye sticks. It's positive for holding on to that first dye. And it does it because we've got these chains of carbohydrates, and these little tiny balls are peptides, proteins, that are linking it, and it's called cross-linking. So we have NAG and NAM that are labeled N-acetylglucosamine and N-acetylmuramic acid. Those are carbohydrates. And then we have pentapeptides. Penta means 5, so there's 1, 2, 3, 4, 5 little amino acids. We have tetrapeptides. Tetra means 4, and there's 4 linking vertically. And when you look at the difference between gram positive and gram negative, the gram negative has the tetrapeptides, but it doesn't have the pentapeptides. We have a direct link between those vertical tetrapeptides. And because of that, it changes how those cell walls will protect the cell. It changes how antibiotics cross the cell. It changes how the dye sticks to them. So when we look at gram-positive bacteria in sort of a side cartoon view, we've got the plasma membrane, we have transmembrane proteins, we have peripheral proteins, and we have a whole bunch of layers of peptidoglycan. We also have this little vertical line called teichoic acid that is not found in gram-negatives. In the gram-negatives, we still have the same kind of membrane, but we have only one little thin layer of the peptidoglycan. We have something called murine lipoprotein, and we have the lipopolysaccharide. So saccharide means sugar, polysaccharide means it's a complex sugar, and lipo means fat. So there is fat attached to a polysaccharide. That is this whole thing here, and that contains the O antigen and the lipid A components. So gram-negatives have a very thin cell wall, but they get this extra outer membrane that is a different way of protecting them. So acid-fast bacteria are gram-positive, so they have the features of the gram-positive, but they also have mycolic acids, and that is a type of wax. And that is a different type of stain that we would use, and it means that wax makes it very difficult for antibiotics to get to them. This is a blow-up of the lipopolysaccharide, always abbreviated LPS. It is a toxin. So lots of gram-negatives that have this create toxins that can affect us. And the one you've probably heard of would be E. coli. E. coli is found in the intestinal tracts of many different types of animals, and if that meat is not cooked well, then the E. coli toxins get into us. Even if the bacteria are dead, the heat has to be high enough to destroy this O antigen. So we have the lipid A, we have the core, and we have the O antigen. And the E. coli that primarily causes the deadly diseases for humans is called E. coli O157H7. And the O refers to this O antigen. What combination of carbohydrates are in that O antigen? And it's O type 157. The capsule is a type of glycocalyx. So a calyx is a covering, glyco means sugar. So polysaccharide, sugar. So this is one type of capsule stain where you see the dark dots are the cells and the gaps around it are where the dye does not stick. And that is where the capsule is, because it's a gooey, sticky layer that the dye cannot stick to. The pili were those really long projections. This is showing two bacteria attached via their pili. And so they are really long. The fimbriae are these shorter projections. And those connections between the bacteria via their pili allow them to exchange genetic information, but also allow them to stick to each other so that when the fimbriae stick them to a surface, they can form a biofilm. The flagella can be arranged in different ways. Trichus means hairy. So monotrichus, you're one hairy, you got just one. Amphitrichus, amphi means both, so the hair is at both ends, front and back. Lophotrichus means you're hairy multiply, but only at one end. And peritrichus, peri means all around, flagella all around. So all of these bacteria would be able to move by flagella, but they're doing it with different numbers and in different positions. We don't have a stain in a light microscope lab where we'd be able to see these flagella, but we will be able to detect if the bacteria are able to move. And they move because the flagella actually rotate each flagella in a clockwise rotation. They will tumble, meaning the flagella spread apart, and it moves in a random fashion. If each flagellum rotates in a counterclockwise direction, then it organizes them, and it's called running. So if we have no sort of chemical gradient difference in concentrations of chemicals, then the flagella are going to rotate counterclockwise and clockwise. No particular directional movement. But if we were to provide, say, a great nutrient source, then the bacteria would probably go in a very run direction toward it. If we provided on a slide something that was harmful, toxic, found offensive to the bacterium, they would run away from it. But if there's no particular good or bad, it's just a neutral environment, there would be a combination of things because the bacteria would move around the slide trying to find the most favorable environment. Now we're going to move on to a generalized eukaryotic organism. So this one has all the features, I think. No, it doesn't have a chloroplast. But not every single-celled eukaryote is going to look like this, but this contains the features that you would find. And so in a very cartoon way, we've got our nucleus, we've got several different types of organelles, and each organelle means a small organ has its own membrane. The nucleus has two membranes, an inner and an outer. The mitochondrion actually has two membranes as well, and when we look at a chloroplast, it has three. This eukaryotic cell is shown with a flagellum. There are lots of protists that have flagella, and reproductive cells often have flagella. So human sperm would be a eukaryotic cell that has a flagellum. So there's lots of different shapes, and far more than in the prokaryotic world. We've got spheres, fusiform, there's bell shapes, they look like little vacuums. There's all sorts of different shapes and sizes, from very small to very large. This is a fluorescent microscope so that you can see in the nucleus there's actually, besides the DNA, there's structure into it, and there is the membrane around it. The endomembrane system, endo means inside again, and membrane. So it's a system of things inside the cytoplasmic membrane. So the nuclear membrane, the outer membrane of the nucleus, is continuous with the rough endoplasmic reticulum. And it's rough because each of these little dots is a ribosome. The smooth endoplasmic reticulum is not necessarily continuous, but it's immediately adjacent to that rough, and it doesn't have any ribosomes. We have the Golgi apparatus, which is a bunch of flat sacs, and then we have lysosomes. Some means a container or a bag, and lyso or lysis means to break. So the DNA and the RNA are in the nucleus. The RNA has to exit the nucleus. The ribosomes, as we're going to see in later chapters, translate that RNA into proteins, and those proteins go through the Golgi apparatus to get modified and finished so that they can exit and do their job, and chemicals, proteins that are needed for digestion of internalized food particles get met up with lysosomes. So there's our rough endoplasmic reticulum with its ribosomes. The Golgi apparatus, an electron micrograph, if we look at it as a cartoon, secretory vesicles means it has enveloped the proteins and other chemicals that have entered it, and it will secrete them out into the cell or to the outside. The peroxisome, so it's a bag containing peroxides. So there's lots of chemicals that can be generated during metabolism that might be harmful to the cell, that would cause oxidation reactions, and the peroxisomes break those down into less harmful chemicals that then can be reused in a cell. The cytoskeleton is the cell skeleton. So there are three different components. There are the microtubules, which are the biggest ones, and it is alternating types of two different proteins that make this big hollow tube. The actin filaments are the thinnest, made out of actin, the microfilaments, so they're very thin. And then our intermediate filaments are several different types of proteins, depending on the cell type, that make this sort of rope. And you can see that the microfilaments underlie, or the intermediate filaments underlie underneath the whole cell, whereas the microfilaments are more attached just under the cell membrane, and the microtubules are more involved in big movements like pushing chromosomes around the cell. So the microfilament is made of actin, and it is found for amoeboid movement, so cells that do cytoplasmic streaming to move, and it's important when we do cell division, and it's part of our muscle contraction. The intermediate filaments, it's like multiple strands of polymerized subunits. They don't get more specific than that because it varies depending on the cell type what type of intermediate filament will be present. Now, where they're found is the same. We've got them in a nuclear envelope, and we get them to stitch cells together, and they vary in their composition because they are a way to identify what type of cell that is. Our microtubules in a molecular model, big hollow tube, and they're involved in moving organelles. So this is sort of a digital image of a microtubule. There are things called motor proteins that will stick to those vesicles that come off the Golgi, and the motor protein will walk it along the railroad of the microtubules. Centrosomes is the whole thing composed of centrioles that are at right angles to each other, and they are where the microtubules are generated. And this is shown in the animal cell. The microtubules are made at the centrosome. They attach to the chromosomes during cell division. A little cartoon of our mitochondria. We have this smooth outer membrane. We have the wavy inner membrane. Anytime you get a wavy membrane, that increases our surface area. There's DNA, there's the ribosome, and the interior of the mitochondria is called the matrix. The interior fluid portion of a cell is the cytoplasm. The fluid interior of a mitochondrion is the matrix. And the little folds are called cristae. This is where the vast majority of our metabolic reactions in eukaryotic cells takes place, inside the mitochondria. The chloroplast has three membranes: smooth outer, smooth inner, and then the stack of little discs. Each disc is called a thylakoid. The liquid inside of the chloroplast is called the stroma. The interior of a thylakoid is the lumen, and if we get a stack of thylakoids, that is called a granum. So it's shown green here because primarily we think about chloroplast and photosynthesis, and then we think of photosynthesis with chlorophyll, and chlorophyll is green. So most chloroplasts are green, but there are things like red algae that have red chloroplasts, and there's brown algae that have brown chloroplasts. It's where the pigments for photosynthesis will be held in eukaryotes. There actually are prokaryotes that can photosynthesize. They have pigments, they just don't have chloroplasts. This eukaryotic cell membrane looks an awful lot like our prokaryotic cell membrane, just that the identity of these peripheral and transmembrane proteins will be different. And now we have a brief appearance by Jackson, the Wonder Chewini. He's getting very restless. So we saw how prokaryotic cells can undergo the plasmolysis or become bloated from being put into a hypotonic solution. Same thing would happen for eukaryotic cells, but then there's also ways of bringing in substances in eukaryotic cells that prokaryotic cells don't do, and it's called endocytosis. Cytosis means cell process, and endo, into. So it's the process of bringing things into a cell. Phagocytosis, P-H-A-G-O, means eating. So it's the eating cell process. And the membrane literally engulfs or eats a particle and brings it in. Some sort of large particle. Could be a food particle. Could be a bacterium. Could be a virus. In pinocytosis, we are bringing in a lot of fluid. It says a small volume, but we're meaning a lot of fluid in that it's more than would pass just by osmosis. So we can envelop and bring in more water than by osmosis alone. And in the last one, we have receptor-mediated endocytosis. It looks a lot like phagocytosis, but there's no receptors here. This is that the cell recognizes something to bring in. Receptor-mediated is we are looking for a very specific something to bring in. It might be a food molecule. It might be some sort of cellular signal that helps the cell to do other processes. But this little fork is the receptor. The molecule is called the ligand, L-I-G-A-N-D. So ligands are the thing the cell wants. Receptors are the things that bind to the ligands, and that triggers the endocytosis to bring things in. The extracellular matrix means it's outside the cell. So this is looking at an animal cell. We've got collagen and we have fibronectin. So they are components that help to cushion the cell surface. Animal cells do not have cell walls. So we need something to protect that plasma membrane. So we have our extracellular matrix. And in the last two slides, we have that generic picture of a prokaryotic cell. So you should be able to go back those slides and review to identify and describe the functions of each of those components. Likewise, the eukaryotic cell. And so I didn't go in any detail at all as to the functions of these things. So that's something to write down in your notes, that you need to know the identity of these structures and what they do in the cell. Don't forget to post your question or comment by 11:59 a.m. on Wednesday, August 26th. And by midnight on that, 11:59 p.m. on that same day, you need to go back in and reply to at least 2 other students to get all of your points and be counted as attendance to not be dropped from the class. If you have any questions, you certainly can email me. I opened a chat so that students can communicate with each other. And I'll also pin a discussion that is sort of holler at me, holler at your instructor if you have a question that you can't find the answer to, or that you can't get the answer from your fellow students. So take care, work on your lectures and lab stuff each week, try to stay on task, and let me know immediately if you are having any kind of technical difficulties. Take care.